I'm Laure Raymond. I've been a clinical pathologist at Eurofins Biomnis for just over 6 years, and I specialize in genetics. Today, we'll talk about the importance of exome sequencing in genetic diseases. When we think of genetic diseases, we usually think about rare diseases. There are also non-rare diseases such as cardiomyopathies. Neuropathies can also be genetic in origin. There are different ways to make a genetic diagnosis. When a patient consults a specialist, the specialist can request the study of a particular gene. They can also ask for a gene panel or a whole exome sequencing, a WES. At Eurofins Biomnis Laboratories, we always perform a WES, so we can have all of the sequenced genes right away and quickly identify the causal variation. Because in certain cases, the specialist won't know what type of panel to request. A WES gives patients a personalized overview of their case. We call this precision medicine. Depending on the pipelines used, we generate up to 30,000 variations in a WES. Based on these variations, we'll apply filters to help identify which variation might be responsible for the patient's phenotype. At Eurofins Biomnis Laboratories, we've been performing high-throughput sequencing for many years, but we started WES in 2016. Of course, before offering it as a diagnostic tool, we carried out a thorough validation of this analysis. We obtain much more information than with a gene panel or single gene sequencing. The most complete information comes from the genome. Then we have the exome, where we study the exons in each gene. Then we reduce the gene panel to a selected number of genes to study the exons. And finally, we focus on the single cell, where we study the exons of a specific gene. We focus on exons because exons are what trigger protein production in the cells, and these proteins allow the cell to function normally. If there's a mutation, a genetic variation, it could mean that a protein is either dysfunctional or missing. And that's why the cell won't function properly, which of course leads to the patient's pathology. When the samples arrive at the Eurofins Biomnis Laboratories, they are handled by a lab specialized in genetics and processing genetic samples. The lab has a team of dedicated technicians, scientific experts, bioinformaticians, and of course, clinical pathologists. Conducting genetic testing in France can only be done in a clinical pathology laboratory. The test must be accredited according to the ISO 15189 standards. The laboratories must be authorized by the French regional health boards to conduct this type of genetic testing. All clinical pathologists must have a genetic certification from the French Agency of Biomedicine. On top of all these regulatory criteria, the quality of the data is also very important. First, we start with a blood sample, which goes through a DNA extraction. It then goes through the capture phase, the exon enrichment phase. Followed by the sequencing phase. This is all handled by the technicians. When the sequencing is finished, the bioinformaticians will transform the sequencer's signals into a signal that can be read by a clinical pathologist. There is also a multidisciplinary team with the clinical pathologist from the lab working in collaboration with the clinicians. We'll select a series of variations that seem pertinent for them, and it's the dialogue with the clinicians that will allow us to reach the most pertinent diagnosis. At Eurofins Biomnis, we feel it's essential that the healthcare provider be included in the interpretation of the results, since they have previously examined the patient. And even if most patient information is provided on the written request, there's always a chance the healthcare provider will remember further details when reviewing the data. We recommend that they directly access the data, so in the same way they would review a patient's imaging tests or biological assessments for more detail, they can also review their patient's genetic data. They can review the data, interpret it, and the lab's clinical pathologist will do the same, and then will compare during video conferences to reach the most pertinent diagnosis. The result can be positive, meaning that we have undeniably identified the variation responsible for the phenotype observed in the patient. Or we can have a variation of undetermined significance, meaning that the gene variation, which could be linked to the patient's phenotype, cannot be positively identified as the causal variation. In that case, further investigation may be made either by requesting additional tests for the patient or running lateral tests of blood relatives. When an exome is negative, it's negative at the exact moment we analyzed it and given the current state of medical knowledge and databases at that moment. But knowledge evolves quickly. Today, some genes are not associated with a specific pathology, but in 2 or 3 years, their data will have been published on cohorts, meaning those genes will be available to be reanalyzed, reinterpreted, because their data was already sequenced. If it's available to be reanalyzed, we can then find a diagnosis. Exome analyses are always being updated. When we have the results, we'll send them to the healthcare provider who will explain them to their patient. The healthcare provider doesn't need to choose between different types of panels. He can just prescribe a WES. We'll do the sequencing, and 6 weeks later, he'll have the complete results. It's important for the patient to have this information and not wait too long to get it. This will allow the patient to get a diagnosis, but it will also significantly impact his healthcare because we can adapt his treatment in order to delay the onset of certain symptoms. It can help in terms of genetic counseling too. Lastly, it can help prognostic impact. Knowing the patient's pathology more precisely allows us to predict its evolution. Eurofins Biomnis Laboratories is really focused on innovation. Bioinformatics pipelines will evolve. Knowledge will evolve. So when we see something new at a congress, we try to validate and apply it to the diagnostic routine so patients can have the most personalized experience possible.